Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay. 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 Yeah, that's great.
Is this going to be mostly your?
No, that's a part of it, but it's going to be, yeah.
Okay.
Or,
Chris. This is not here. Okay. All right, video land, please roll tape. It's my pleasure to introduce Joseph Bockhorst. He is a graduate student at University of Wisconsin-Madison. We met him at NIPS just a month or so ago, and he had a great poster there. And he's done lots of other interesting work in computational biology and machine learning. And we'll talk about that.
Thank you. Thank you for inviting me. I'm happy to be here to discuss some of my research. So how is the information encoded in DNA molecules used to construct complex organisms? This is a problem that has intrigued scientists ever since the discovery of the structure of DNA over 50 years ago, and it's certain to keep them busy for well into the future. And great progress has been made in the last 50 years, definitely. But I think that we find ourselves now in a moment where the combination of high throughput technologies and powerful computers, because of those two things and others, that there is a potential for very exciting developments in the near term. And I believe that because of the tremendous amount of data that needs to be analyzed and the complex relationships and uncertainty involved, that machine learning and probabilistic methods will have a vital role in biology and medicine in the coming years. So today I'm going to talk about research that I've done on using machine learning and probabilistic models to discover important elements that are encoded in the DNA sequences of bacterial genomes. Okay, so thanks to advances in high-throughput sequencing technologies, we now have the complete genome sequence of 250 or so genomes, and this number is growing, and it's sure to grow in the years to come. And from a computer science point of view, the genome sequence can be thought of as a very long string from a four-letter alphabet with A, C's, G's, and T's. So now that we have these genome sequences, what some folks have started to look at doing is to try to identify important subsequences or elements, regulatory elements, that play some functional role in the cell. And some of these elements are known from wet lab experiments, especially in heavily studied organisms. So after we get the elements, then what other folks look to do is to combine, or to try to figure out how these elements interact with each other in networks. So here I've shown a metabolic network and a gene regulation network. And some of these are known as well, these interactions. So we're in a situation now where we have some things that are known, and we have this very important problem. And this is a setting where machine learning is likely to have, or potentially can have a very important impact. So our lab has looked at both of these tasks, learning the elements and learning the networks. However, my research is only focused on the element sides of things, and that's what I'm going to talk about today. So the task that I'm going to be talking about is the following. So I'm assuming that we have the complete DNA sequence of a bacterial genome, and we have some known elements that we're going to use for training, and I'm going to talk more about exactly what these elements are in a second. And we also optionally have some experimental data that we can use as well. And then our task is to use machine learning methods to learn a probabilistic model, and then to apply this model to predict a parts list of all the regulatory elements in the genome. So we have some, and we want to predict them all. That's the task. So the overview of the talk today is I'm going to begin by describing contributions that our research has made in computational biology. And I'm specifically going to describe probabilistic models for operons, promoters, and terminators. These are three of the elements that we're trying to discover. And then after I've talked about that, I'm going to get into algorithmic contributions that the work has made in machine learning. I'll talk about a method we've developed for detecting and modeling overlapping elements in sequence data. And also I'll talk about a method for refining the structure of stochastic grammars. So I'm going to begin with a bit of background. This is the biology background for the talk on transcription in bacteria. So what I've shown in this slide here is the horizontal bit is the thing that looks like a ladder on its side is a long, it's meant to represent a long string of DNA. And in this DNA sequence, there are genes. Genes are subsequences of DNA that code for proteins. And the process of creating a protein from a gene, the first step in that process is called transcription. And that creates a molecule of RNA, so that the downward arrow is meant to indicate the process of transcription. And for the purposes of transcription in bacteria, genes are grouped together into operons. So here's an example where we have two genes that are grouped together in an operon. And what an operon is it's a sequence of contiguous genes that are transcribed as a unit. So here's an operon that has two genes. So in general, an operon could have 1, 2, 3, or more, up till 10 or more genes. So one of our tasks are going to be to develop models for predicting how genes are grouped together into operons. Okay, so the process of transcription is controlled by two important signals. At the beginning, there is a signal which is encoded in DNA, which is called the promoter. This is the start transcription signal. And then at the end is a signal called the terminator, which indicates where transcription stops. So we're going to be also interested in finding these elements as well. So we're going to be talking about developing probabilistic models for recognizing operons, promoters, and terminators. So we've developed models based on Bayes nets for modeling operons. And in this task, then, we're given a set of contiguous genes, and the decision is, do these genes belong to the same operon or not? So we've been working on that for a little bit. And more recently, we've also developed a unified probabilistic model, which is based on probabilistic sequence models or hidden Markov models or stochastic context-free grammars. It's a unified model of promoters, operons, and terminators. And the probability distribution that these models represent is they represent the probability. The prediction is to find a probability of some set of elements given a DNA sequence. So we're given an input sequence of DNA, and the prediction task is to find where the elements are. So we have this probability distribution over elements given sequence. And the way this is done is that we have the components of our model, or the model itself, represents this likelihood of a sequence given the elements. And we're going to have components for all of the different elements that we're interested in predicting. And each of those components are going to represent a probability distribution of a DNA sequence for this corresponding element. Okay, so this is the model architecture of this unified model for promoters, operons, and terminators. And I'm not going to have a chance to go into the details of all these different components here. But I'd like to point out a few things. First, the different shapes of the models are meant to indicate something about the probability distribution for the bit of sequence that component represents, or that component models. So let me just say one other thing, because I kind of missed it on the last slide. That green is going to mean promoters. That's where transcription starts, and red is going to mean terminators. So that's going to be something that is going to kind of come up So I should have mentioned that when I defined those before. So we have the components of a terminator, and then the components of a promoter are over here, and then we have components of a terminator over here. And each of those different shapes has a probability distribution associated with it. And we connect them all together in this fashion. And this has been, we hand designed this model in order to model what is known about these elements. So I'm going to just drill down a little bit on this component here for the triangle that says stem loop and look at our specific representation for this. So this is based on, we use a stochastic context-free grammar for this. And the reason why we use a stochastic context-free grammar instead of a stochastic regular grammar is that there are long-range dependencies between positions in terminators that we need to capture that cannot be captured very naturally with stochastic regular grammar. So what I've shown here, so if you want to read the sequence off in a left to right fashion, we'd start here, read up and around, and then down there. So the yellow dashed lines are it's a property of the terminator, and what those are meant to indicate is there's a dependency between those positions that are connected. So we use a context-free grammar for that. And here's the structure of it. I'm not going to be able to talk about it in much detail, but this was hand designed for modeling terminators, and a little bit later I will speak about algorithms for actually refining the structure or learning the productions and non-terminals of a stochastic grammar like this. So we have these. What's that? I'm referring. Yeah, this. That's right. So that's, yeah. So I've written this as a DNA sequence, but the important part of what makes a terminator is what happens when it's transcribed. But we're still recognizing these things in DNA, so if you just think about these dependencies. Right. Okay, so we have the non-terminals on the left-hand side, and the productions for each of these are shown on the right. And the terminals in this grammar are just A, C, G, and T. And if we just look at one of these, so this is shorthand for a whole set of productions where B, so we have B, L, and BR. This is just the generic, or a shorthand for any of the bases, any A, C, G, or T. So we can expand that out into 16 different productions. And each production has a probability associated with it because this is a stochastic grammar. Okay. So the learning tasks that we have to learn for these models are, if we're assuming the structure is fixed, the learning task is to set the probabilities of these models. And what we are given is a set of partially annotated sequences. So what we have, for example, here's a training sequence. You may not be able to see this, but it's not important, the specific basis, but this is a long string of A, C, G, and T, and we might know that this box is located here. So that's going to give us the information that we're going to use to set the probabilities, and we use a standard expectation maximization to set these probabilities. And so then, if we have a fully trained model, when we're applying it, what we do is we're going to use the Viterbi algorithm to find the most probable parse. And what a parse is an alignment of subsequences. So we're given an input sequence, and we want to find the element. So we're going to make an alignment of subsequences to the components of the model. These shapes are the components. And the property of this alignment is that that every position in the sequence is aligned to exactly one component. Okay? And we're going to relax this a bit later on when I talk about overlapping elements, but I want to just point out that that every position now is only associated with a single component here.
Are you getting the structure of this model from somewhere, or is this a hypothetical if you have the structure?
Mm-hmm. So the model structure is designed. We designed it by hand to try to capture. Right. Yeah. So this is nothing that if you read an introductory textbook on genetics, that these things would kind of naturally fall out. Okay. So up until now, I've only talked about how our models explain DNA sequences, but there's other types of evidence that we would like to be able to use to make predictions of these elements. And one example of this is information that comes from the results of gene expression experiments. So what these expression experiments measure is they measure the transcription activity at specific positions in the genome, which are called probes. So here I've shown, here's a piece of DNA, this yellow string, and then we might have probes at 3 different positions. So what that means is we can measure over a set of different conditions the activity level of how actively that position is being transcribed. So this is going to be important because this is what we're trying to find, where are the boundaries of transcription. And so if probes are in the same operon, there's going to be this correlation between their expression patterns. So what we do then is we, in addition to our DNA sequence, we're going to construct a sequence of expression correlations that are going to be aligned to where we have probes, and we're going to then, our models will be able to give us the probability of elements given both sequence and expression. Okay, so how well did these models do? And I'm going to look specifically at how accurate are our predictions of operons. And I'm also going to, we're interested in what is the value of using multiple sources of evidence, both DNA sequence and expression. And we'll have three models that I'll consider. Number one is the sequence plus expression unified model, which is the one that I've just described, which is this unified model of promoters, operons, and terminators. And second model will be a model that uses sequence and expression but only predicts operons. This is based on our earlier work for predicting operons. And then lastly, we'll use operon prediction methods that are based on just the sequence only, that only look at how far apart the genes are. It's based on the spacing between genes. And this is, had been the standard method for predicting operons. So this is ROC curves for each of these methods. So let's see. On the bottom here, on the x-axis, we have the false positive rate. That's the probability that something that is actually false is miscorrectly labeled as true. So we want that to be 0. And on the y-axis, we have the true positive rate, which gives the probability that something that's true is actually called true. So we want this to be 100%. So ideally, we're at 0, 100%. And the intergene spacing is the green line. The sequence plus expression operon only model is red, and our unified model is this yellow dot. So we're able to, in these two cases, we're able to come up with, given a candidate operon, we have a probability that it is a true operon. And by varying the threshold at which we separate true from false predictions, we're able to get this curve. So we can see that that over the whole range of false positive rate, our sequence plus expression operon only model outperforms the intergene spacing. And also that our unified model, although it's addressing a more specific problem of predicting promoters, operons, and terminators, appears to do just as well or maybe even slightly better than a model specifically designed for predicting operons. Yes? Okay. Let's go back then. So we're going to be, at each of these positions where we have these probes, we're going to get a correlation. A correlation, which is the correlation between the expression profile at the probe and the expression profile at a neighboring probe. Okay? And so we can align these correlations to specific positions. And we also have a model for the correlation, for correlations that are between probes that are in the same operon. And then we have a model, a probabilistic model for the correlation between probes that are not in the same operon. I don't know if it makes it easier to run the Viterbi algorithm.
The idea is that you don't have to probes to be locked within
Oh no, it's providing another bit of evidence that we can use to decide where to actually perform this parse. So maybe it was a little bit misleading that these probes locked you into this. But they're another piece of evidence that we're going to use when we're parsing a piece of DNA that's aligned to these measurements.
Okay, so in my head, I have this big state diagram of different things that we're doing. I don't see where, are you emitting
Okay.
flow?
Yeah, so we're emitting correlations. You can think of it like that.
Well, I think one thing I don't, I'm not sure which of them Max is confused about.
Okay.
I'm only confused about one of them.
Okay.
Right, so one question is, what do the probes do? And my understanding of that is the probes are in the same operon,
Mhm.
they tend to be pressed together, so there's operons, they tend to be pressed apart, so we can get some posterior. The thing I'm confused about is you essentially have two independent kinds of information.
Mm-hmm.
Maybe it's stochastic, right? So one is you've got this nice model of elements of elements in a DNA sequence, and then you have this other model, and that whenever you have two different probability estimates,
Mm-hmm.
there's the question of how do you combine them.
Mm-hmm.
So that, right? You basically
Right.
have two independent sources of information. How do you
Right.
combine those two?
Right. Okay. So so we make a conditional independence assumption that the sequence is conditionally independent of the correlation from the probe, given the state that it's aligned to. Okay. So then within each of these things, before this state would have a probability distribution over just the sequence part.
So if you can get this whole kind of probability together, what will happen?
Yeah, if I wanted to compute the likelihood of a sequence, and if I want to compute the likelihood of this bit of sequence and this correlation for this probe being emitted by this state here, I'd multiply those together. Yeah. Mm-hmm. Mm-hmm. Mm-hmm.
Then you would still have a reduce your imbalance because if you don't have enough probes, then the sequence data might overwhelm the sequence. But it seems like you're doing things a little different here because you said your measurement is not the probe expression level
Yeah.
itself, correlation. You're looking at correlation
Right.
between neighboring probes.
Right. The reason we do the correlation is so we can make this independence assumption. And if we didn't do correlations, if we just emitted the raw measurements, now there's going to be correlations that you can't make this independence assumption because if they're in the same operon, they're transcribed at the same level. Okay? Does that make some sense? Then we'd have to remember what that level is for all of the probes that are in that operon. But now we're just going to take the correlation and we're going to assume that that measure is going to... Well, actually, okay, so we have a probe here, and we're going to have where the gene boundaries are. And we get an expression measurement for the gene. And we take the correlation between the profile at the probe and its neighboring gene. Okay? Because genes are always in some operon. That's, yeah. Yeah, it'd be the genes to genes are one part of it, and also we have these probes that are in intergenic regions, and so then we would take these probes that are in intergenic regions either could be in some operon or not. They could just be in regions that are never transcribed. Okay, so we take the correlation between the probe in an intergenic region and then it's the nearest neighbor gene, and we can do that in both directions. Okay. Okay, so this is, we're getting near the end of this first part where I talk about this computational biology contributions. But just to summarize what I've talked about so far is that I talked about this method for predicting regulatory elements in bacterial genomes. And so along with the Blattner lab at Wisconsin, who were the ones that published this E. coli genome sequence, they've been generating expression data, and we've collaborated with them to predict a complete map of all the operons, promoters, and terminators in E. coli. And we hope to apply this to other elements or other bacteria, Xenarobis and Rotobacter, soon. And so we've come up with this model, and we think it's novel for a couple reasons. First of all, it incorporates multiple data sources where lots of previous methods just looked at sequence or just looked at expression, but it also can make predictions with what's available. If you want to apply this to a genome that you don't have any expression data, that you can go ahead and do this. And also that most of the previous methods just looked at predicting operons, but we're looking at this more specific operons, promoters, and terminators predictions.
Can we go back to the first slide?
Yes.
So I'm sorry, you said that it was a state back, but we could also put state there.
Oh, the green. The green, yeah. So
So now let's go back to your model, right? So it looks like you're being spacing gets you off this part.
yeah, that's very good. I mean, for as easy as it.
So how did you? So where's the interesting spacing in the model?
Okay. Right here.
Okay, and what do you? How do you? So that looks like that's the most important part of the model. So how do you actually model that?
So we use a semi-Markov model here.
What is that?
So that means that we have a probability distribution over the length of the sequence, and then we have the probability distribution over the bases, and it's homogeneous for all the positions in that intergenic region.
Okay. What is the actually for that?
It would just look at the spacing. It would just ignore the specific composition of the bases, and it would just look at the spacing itself. Right. Right. Yeah, that's. I mean, it's.
So basically, so you're capturing. Are you capturing all the information that's previous all captured plus more, or is there anything you really have doing this way?
Well, we use a little bit that we don't have. So this we have. This is the intergenic region between genes that are in the same operon. This bit here. So for the intergenic region between genes that are in different operons, we don't only have a measure for a distribution over that distance, but it's kind of encoded in the distances that are in all these other states. Does that make sense? That this, even though that's we have all these other states that they all have length distributions, so it kind of combines into some distribution. We don't explicitly take advantage of what the statistics were between genes that are in the same operon. Yeah, you could like assume the most likely. Right, right. Yeah, that's a good idea because I was concerned about this losing this ability, and I was thinking, do we need to try to fit this in some other way? Yeah.
Are there any differences between the distributions within an opron and cross opron?
Yes. So that's what the previous model was trying to capture with just those differences.
Not differences in the length, but
Oh.
the actual base pair.
Yes, that is because if you're in between, if you're in an operon, you don't need all these other signals here. You don't have these things.
I'm talking about the intergenic region.
Mm-hmm. But maybe if I go back, I don't know if I should.
Or maybe we should go on and I don't know.
Well, I've come. So if we're in, here's an operon. Here's the intergenic region here. It doesn't have any of these signals, these promoters or terminators. So it's not going to have the sequences corresponding to those. So the.
So there's definitely a difference between the different antigens now, or
Are a lot not likely to anyhow.
I guess what I was asking is, so okay, so we've got that particular, right? So we've got those intergenic regions within the opron,
Mm-hmm.
and we've got intergenic regions between opron.
Right, like this one here.
And so the previous people were doing everything just by measuring the length of those.
Mm-hmm.
And the question is there any more information, those maybe between genes, there's old genes that degraded or something, ends that degraded or something. Maybe they somehow look more. Is there anything? Does anyone look at whether those base pairs somehow have different patterns? They're all not photos.
Okay, just to look at I mean, like the gross statistics of base composition. I mean, they certainly have different patterns because if you're in between genes that are in different operons, you're going to find these elements which are encoded in DNA. Yeah, I don't know what. I mean, I would expect there to be differences because there's other things that are in there in between genes in the different operons that aren't found in between genes in the same operon. Not right now, no. Yes. They're variable, and there's different types of promoters, and every individual type of promoter also has variation among the specific composition. Yeah, it's not trivial just to find those things. Yeah, so you're kind of right. So if we were trying to make gene predictions along with everything else, then we'd have to look at all these different reading frames. But the model I'm describing, we're kind of assuming that the genes are there and predicting the other bits.
So, but then you know that these overlap. You don't take enough space into account that the one also on the terminator, you can have a terminator further away, but we can't apply to one point to the other.
Well, the terminator only has to do with transcription. So I think you're thinking about translation ending. But the termination would apply to transcription. Well, the simple answer is that we just did this Viterbi path to get these predictions. But we could, so we just did the Viterbi, so we just had either yes or no for each of these. But yeah, that this is something I'm working on is making this yellow curve go all the way up and down too. Okay, so if there's no more questions, I'll move on to the second part where I'm going to talk about specific algorithmic contributions that our work has made. So now I'm going to talk about a problem with standard sequence models, probabilistic sequence models, is that they don't naturally handle overlapping elements. And this is an issue in our case because elements in bacterial genomes frequently overlap each other. So here's an example. This is a real case from E. coli where the promoters, which are the green triangles, you can see that they overlap each other. They overlap genes. Terminator overlaps a gene a bit. And this isn't a special case. These kinds of things happen with some regularity throughout the whole genome. So I'm going to talk about this model that we've developed to be able to account for this. And we think that it's a general approach that can be applied to other domains that have sequence data, or perhaps two-dimensional or three-dimensional data that also have overlapping elements. Yeah, I don't know. I mean, that it's certainly, I don't think it's trying to optimize that, but it is. There is more space pressures in these small bacteria, and being able to put things to encode them using this sort of like compression or something like this. Okay, so before I talk about what we did, I'm going to talk about a few alternative approaches to dealing with overlapping elements. So the first way to do this would be to not deal with it, and that's sort of what the approach I've described previously based on HMMs, where we just don't represent or model these things. Another approach would be to break the HMM apart and just take our individual the components of the model for the individual sequence elements and make predictions with these. So what we'd have, if we have components for these individual models and we're given an input sequence, we'd scan these along, make predictions along the length of the sequence, and then we keep the best ones. The problem here is that we've lost this relationship among these elements. A third approach would be to expand our grammar or Markov model to have states or non-terminals for cases of overlap. So here, just shown an example where here we have a state for a gene. That would be in the standard model. Now we'd have to also consider it when it overlaps a promoter by a bit more, and so on. The problem here is that we're going to need the number of states we'll need will grow exponentially with the kinds of overlaps that we allow. So then the other approach is what I'm going to talk about is based on changing the sample space over which we define distributions. So this, I've tried to illustrate how we do this on this slide. So here's an example where we're trying to make predictions for a length 8 input sequence. And for the standard HMM approach, it assigns labels to each position. Okay? So if this is a length 8 input sequence and we have a green thing at 2, a blue thing from 4 to 5, and a red thing at 7, that the event in the sample space corresponding to those predictions is represented here. We have a green label at 2, blue label at 4 or 5, and a red label at 7. So for our approach, instead of putting labels on on positions, we put labels on subsequences. So here's a configuration of predictions that are overlapping, and here's the representation of those predictions in the sample space by the models that I'm talking about now. And so each square here represents a subsequence of the original sequence. And we've shaded this one blue because this is the subsequence that starts, or this one green. This is the subsequence that starts at 4 and ends at 5, which corresponds to that prediction there. So that's the sample space we're going to use. And now I'm going to talk about how we define a probability distribution over assignment of labels to this. Okay, so here's how our approach, how we use this approach. So we have individual element models for each of the individual elements we want to find. And given an input sequence, we're first going to scan through it with these individual element models to make candidate predictions. And then we're going to construct a probabilistic model, which is based on Markov networks or conditional random fields, and perform inference on this model to decide what to accept. Okay, now I'll go into a little bit more detail on the structure of this model. So this model has a single variable X, which corresponds to an observable input sequence. So in our case, this is a DNA sequence. And we have a set of output variables, which are the Y's, Y1 all the way through YL plus 1, if L is the length of the input sequence. Okay, and what these output random variables, the values that they take are the predictions. Okay, and we're going to impose, so what the value of YA is the 8th match of a configuration. So a configuration is the set of predictions associated with an input sequence. So if this is the input sequence here, the configuration will be the collection of all those, all the predictions for one input sequence. And so we're going to impose an ordering over subsequences. So now we can say what the first match is, what the second match is, and the third, and so on. And so the value that YA will take on is the 8th match. So if we have M things in our configuration, here the value of Y1 is the first one, the Y2 is the second one, and so on to YM. And so the remaining output variables will just be set to special null values. This helps us deal with the fact that we don't know beforehand how many elements there are encoded here. And for the task that we've been applying these models to, we've made an additional assumption that is not doesn't hurt us in any way in this problem, that no two elements or no two matches start at the same position. We're allowing overlaps, but we're not allowing them to start at the same position. So if we do this, now we can limit the total number of matches in any configuration to the length of the sequence. So we can get away with just L output variables. Okay. Yes?
So Y1, the dynamics of Y1
Yep.
green starts in each one?
Yeah, if this is the values that it takes on. No, it doesn't start at position 1. These are, we've set the value of Y1 to be a green thing that starts here and ends here. And that doesn't have to necessarily mean that it's position 1, but it's just the first element in this configuration.
So that the starting and ending point is defined by the pre-processing step of finding
So the pre-processing step, we can think of it as scoring all possible placements for green. Okay? Now, let's say that the first element begins at position 50 and ends at 88 or something like that. Then that's what I'm calling a match, is a match of the green thing to position 50 to 88. And if that's the first one, then that matches the value of Y1.
So the first is the leftmost match.
Right.
Okay, so Y1 has the value object, start, end.
Right.
And these are sorted by left position.
That's how we sort it. Right. We sort it by starting position because we're assuming that no two start at the same spot. So now we can have this ordering over any of these configurations. Okay. So that's the structure of this Markov net. And in order to define a probability distribution for the output variables given input, we have to talk about the cliques and the clique potentials. And I just want to point out what the cliques are in this slide here. So the cliques in this graph are these triangular things. And we're going to say that clique QA is composed of X, YA, and YA plus 1. So there'll be L cliques in this graph. Okay, and here's a representation of the probability of a set of predictions or assignments to Y given an input sequence X. So we have a product over all of our cliques. So we go from A to L. So that's over all our cliques of these two terms, G and H. And G is what we call the match function. And what it does is it measures the compatibility between a subsequence and an element type. It only depends on the subsequence matched by that element type. And this is based on our the individual element models we have for the different types. Okay? And then we have H, which measures, we call it the interaction function, and it measures the compatibility between adjacent pairs of elements. So these are kind of like the transition functions of an HMM, but the key is that that these adjacent pairs of elements may overlap each other. So we're going to have a score that depends on the types of adjacent elements and also their degree of overlap, or the amount that they're separated by. Okay. So let me talk a bit about inference now. So the default, if you just use the belief propagation algorithm to perform inference, it would be L to the fifth. But that would be, in our case, too much. So we've developed an inference technique that exploits some of the properties of this interaction function in order to come up with a L squared algorithm. And so the first property that we exploit is that the value of the interaction function is invariant to the start. So here we have H depends on two adjacent matches, YA and YA plus 1 in the sequence. The value of H is independent and invariant of the start position of the upstream element and the end position of the downstream element. What that means pictorially is if we have two arrangements here, we have a green triangle upstream of a blue thing separated by 19 in this part of the DNA sequence, and we have a green triangle upstream of a blue thing that's much bigger down here that's also separated by 19, that those will have the same value. Okay, so that's one of the properties. And the other thing is that even if the difference in these two things, so here we have this one's 19 and this one's 18, if it's just off by 1, because of our representation, this is often going to result in having the same value. Okay, but for some cases, of course, it won't. Otherwise, it would just have the same value for everything. So maybe if we group things into groups of this spacing into groups of 10, then maybe if they were separated by 20, they'd have a different value there. I'm not going to go into any more detail on this inference measure, but if anybody's interested, talk to me afterwards, and I'd be happy to go over it. Yes, Max.
Curious about the independence assumption. Usually you have a green triangle that overlaps.
Mm-hmm.
The green triangle is related to the next one, right? So what's important is just that triangle two away, or is it the fact that if you overlap, it has in order to effectively overlap?
Well, it would actually affect both if these genes are its neighbors. because its upstream neighbor would be the gene that it overlaps, because we go by the start position, and its downstream neighbor would be the next gene.
Okay, so I have green triangle starts at position 1.
Mm-hmm.
Another gene starts at position 2.
Oh, I see.
It overlaps a little bit.
Yeah.
And then there's a space, and then there's another gene. I see the green triangle regulating the second gene first.
Yeah. That would be a problem. Although that, fortunately, doesn't happen where it would, in that type of overlaps. But there certainly is some. We're making this assumption where things only depend on its neighbors, and this is done to be able to make these algorithms efficient. But there could be a case where we have things overlapping each other, and it's not even dependent on something else that it overlaps because of there's some intervening element in between. Yeah. All right, so we're going to compare 3 ways of making predictions of promoters and terminators. And so I don't think I said this earlier, but all of these experiments were done on E. coli, and this is all test set accuracy cross-validation, tenfold cross-validation. So we're going to compare 3 approaches. The Markov network approach I just talked about, our unified promoter, terminator, operon HMM I talked about earlier. So this one represents dependencies between elements, but it can't deal with overlaps. And I'm also going to look at scanning. So if we break the HMM apart and just do the scanning approach, how well do we do? So here are precision recall curves for these 2 tasks. On the left, we have the task of predicting terminators, and on the right, we have the task of predicting promoters. So we have the x-axis here is recall, which is the total number of true positive predictions divided by the total number of true examples. So how many of the things that are really terminators did we find? And then we have precision, which is of the things that we predicted to be terminators, how many of them actually are. So this is slightly different than the ROC curve that we'd rather be up here at 1, 1. But at any point along recall, the higher up you are, the better. So we have our Markov network approach is the yellow line, and the unified HMM is the red line, and the scanning approach is green. So we see that the scanning doesn't do well at all. So there's clearly, it's important to be able to model these dependencies among these elements. And we see that our approach, the Markov network approach, does better on the terminator task, but on the promoter task, it's sort of a wash where it doesn't do as well at low recall range, but it does better at high recall ranges. Okay. So that ends the bit on overlapping elements. Now I'm going to talk about, I'm going to quickly describe methods that we've developed for learning the structure of stochastic context-free grammars. So this was our terminator grammar that I talked about a bit earlier. So we have sequences where we can, training sequences that can be used to estimate the probabilities, but what if our initial hypothesis of this structure is wrong a bit? Can we find a way to learn this? So the task then for that I'm going to look at for SCFG refinement is that we're given a grammar, a stochastic grammar with probabilities, and along with a set of training sequences. And what we want to do is we want to adjust the CFG structure and then relearn the probabilities. Okay, so the key point of our approach uses this idea of contexts. So what I've shown here is 2 different contexts for the non-terminal W. So these are little bits of a parse tree. Okay, so here's the non-terminal W on the left that came from V via the production AWT. And here's a terminal W on the right that came from V by the production CWG. Okay? So every non-terminal in a parse tree exists in some context. And what our approach does is we gather statistics of how non-terminals are used in these different contexts. So we do this by counting over our training sequences. And so what this is meant to represent is that, so if here's W in this context, and W, let's say, has 4 productions, we count the number of times it is used in the first, second, third, and fourth cases. And these, I put observed in quotes because we don't have fully labeled data. We don't have parse trees. We just have sequences. So these are more like expected counts. Okay? So we do that for all of our different contexts. So all the different contexts for W and all of our other non-terminals that we have. And the thing that we look for then is we look to see if a non-terminal is used in some context very differently than it is used in all other contexts. Okay? Because this is a context-free grammar, so we would expect that the statistics for W being used in this context to be the same as W in this context here. Okay, so here's the case where these two are not the same. It's not the raw numbers that are important, but it's just the distribution. So this one looks like it's giving preference to the middle two productions, but over here we're giving preference to the first and last productions. Okay, so if we find such a case where we have a context that's being used very differently from all other contexts, we're going to introduce a new non-terminal to deal with this specific case. And then we'll retrain the grammar, retrain the probabilities of the grammar given our refined structure. So what we want to look at for this method is how does the accuracy of our grammar change as non-terminals are added? And we're going to look at a task of predicting Terminator sequences in E. coli. And we're going to look at 3 different methods for adding non-terminals. The first 2 are based on a heuristic, based on this idea of looking for these places where the contexts are used differently, or where a non-terminal is used differently in one context than the other. And we're going to look at 2 different ways to determine what the difference is. One is based on a chi-squared test on the counts, and another is based on the Kullback-Leibler divergence of the frequencies that we would get for each of these different contexts. And then we also have a random control that just randomly chooses where to refine the grammar by introducing new non-terminals. So here's this graph shows the area under the ROC curve as we add additional non-terminals. So we have additional non-terminals down here, and the y-axis is the area under the curve, so we want to be at 1. And if we just randomly guessed, we'd be at 5, or 0.5, so that we start down there at 0.5. So we see that randomly adding new non-terminals doesn't seem to help in this task. But both the chi-squared and KL divergence heuristics, which makes targeted changes to the grammar, seem to improve accuracy for a little while. Okay, so I've talked about 2 specific algorithmic contributions that our research has made into machine learning. I started by talking about dealing with overlapping elements in sequence data. And we've developed models that can represent both the specific properties of elements as well as dependencies between them. And as an HMM can do, but also can deal with overlapping cases. And then I just talked about an algorithm for refining the structure of a stochastic context-free grammar. And our approach is based by diagnostically figuring out where to introduce non-terminals. And this contrasts to previous work in grammar induction. So I should say that, so in order, the cost of adding a new non-terminal is that we have to retrain the probabilities. We have to do a training cycle. So the other methods that look at refining grammar structure require retraining every time it evaluates an operation. It has to say, if I made this change to the grammar, what would the likelihood of our data be? And so then there's all this retraining that needs to happen, and it's expensive if you don't have fully labeled training examples. No.
Very simple. They started from just a random assignment of non-terminal they can start with just random rules, and then just run the wasn't at all incremental.
You just did this over and over again, or something?
No, they just did it once.
Oh.
You just start with a random set of the random grammar.
Mm-hmm.
And so Spolky, I don't know if Spolky first time was trying to actually. So there's different ways of evaluating it. From the point, these guys Spolky and Ken are both natural language processing guys, right? So
right.
One of those stuff that match the actual, what they call it the actual structure language. But the very intelligent results were actually pretty good, not good from that point of view of matching the structure of language, but were fine in terms of minimizing metrics,
Okay.
which is probably the most relevant criteria to use.
So am I meeting with you today at all?
No, but
Okay.
probably very much like that.
Okay.
But Ken was in my, I was in the same office where the 1094 was.
Okay. Okay.
This is the only private talk I can
Okay. Sorry.
say. No, I'm kidding.
Okay, so I talked about two different contributions that this work has made in machine learning, but we've been working in other areas. Semi-supervised learning, we've been looking at ways to exploit weakly labeled training examples, and also recently I've got into multi-relational data mining a bit. And I'm not going to talk about these, but if you're interested, I'd be more than happy to talk about this with you afterwards. So a bit about where we're going currently. And everything I've talked about up until this point has been making predictions on a single genome. And we'd like to apply our models to other genomes to make predictions, but we'd also like to be able to use the relationships among genomes to make more accurate predictions. So what I've shown in this example is that we have these two genomes, say genome 1 and genome 2, and the task is to decide if these two genes are in the same operand in genome 2, and if these two genes are in the same operand in genome 1. And if these genes are related evolutionarily, that the value of those predictions are going to be correlated to each other. So what we could do then is if we have experimental data on one of the genomes, let's say microarray or gene expression experiments from one genome, then we can use evolutionary relationships to be able to propagate evidence back to make better predictions on the other genome. So this is where work I'm currently working on right now. Okay, so I've talked about using machine learning and probabilistic models to solve important biological problems, and I think machine learning has a very important role to play in biology and medicine in the coming years. So we have these important problems, and we have complex relationships such as evolutionary trees, and we couple that with high throughput technology and uncertainty, and I think that there's, again, a great potential for machine learning and probabilistic models here. And this will make all machine learning researchers happier, as well as contributing to society at large. I'd like to thank my collaborators, my advisor, Mark Craven, and David Page from Biostatistics and Medical Informatics. In computer sciences, Jude Chavlik has been a great help and always ready to answer my questions. And I've also worked with Irene and Keith. And from the Blattner Lab, we have Fred and Yu-Q and Ming-Zhu, who have been available for countless questions about biology. And Nicole and Jeremy in the Perner Lab have been very useful with helping me understand evolution. And also like to thank my funding sources. And thank you. Questions, anyone? Okay. All right.